So I looked at bacterial growth and the implications it might have for space exploration, where obviously in space exploration you have to keep everything very sterile for equipment function as well as other things we'll get to later. The first thing we did is we looked at to see how much bacteria is actually around us. We found that a lot more than you might think. So this is bacterial colonies grown from a cell phone, and each one of these little yellow dots right here is actually thousands to millions of little bacteria all grouped together. And this is some bacteria colonies grown from human skin. That's healthy, normal human skin. As you can see, there are a lot of bacteria. But luckily we have all these cleaners which work to fight the bacteria. But what we found was they're not as effective as you might think. So even though they all claim to be 99.99% resistant, they're not necessarily true for all types of bacteria. Types of bacteria we looked at, only 3 bacterial or antibacterials were actually efficient at combating them. As you can see indicated by these little halos or zones of inhibition where bacteria can't grow because the effect of these cleaners. And then what this might have for space exploration. Does you have a question?
No.
Oh, in terms of space exploration, like I said, the equipment needs to be kept very sterile in order to have the equipment function properly. I was speaking with a guy down at a Goddard Space Flight Center in Washington, and he was saying how the 2 biggest issues he has to deal with are microbes and dust. I mean, because the precision for these equipment is so precise that even a small microbe would disrupt the function. And also in terms of astrobiology, something like the Mars rover here, it's looking for microbes on the surface of Mars. And it needs to know to make sure that it does not have any bacterial contaminants. So if it finds something, it knows it truly is Mars life and not Earth life that it transported. And also there's the other issue of you don't want to contaminate a foreign planet, even if it's not in relation to your data, but if there is any kind of ecology on a planet like Mars, which there might be, data is indicating that it's actually possible and in some cases probable to have life there. You don't want to damage that with our bacteria. As you can see throughout history and through ecology, anytime you take microbes from a place where they don't belong and put them there, you end up disrupting the ecology to a great extent.
Right.
So,
what was the most effective cleaner?
The effective cleaner we found was ETOH or ethyl alcohol. It's a laboratory cleaner. We used 70%, and it seemed to be the most effective we could find.
What were some of the other ones that you compared? Are there any others out of this picture?
Yeah, so we have isopropyl alcohol was pretty efficient. Windex multi-surface cleaner actually was surprisingly efficient.
Cool.
And Dial hand soap, looking at a hand soap, was the most efficient we could find on the market. So
What about Mr. Clean?
Mr. Clean, not so much. Mr. Clean, yeah, it was unfortunate. Yeah, that's what we thought, because these are all claiming the same thing. And I thought Mr. Clean, but again, that's just for the SA and PA bacteria we used. So if we use different bacteria, we'd get different results.